Hey guys, how's it going?
Good.
All right, I'm trying to remind myself of where we were last time. Are there any questions about what we've studied so far in terms of thermoregulation? Once again, the goal of this section of the material is kind of so you understand what the options are for organisms that have to regulate their body temperature, not through the production of metabolic heat as the primary source of body temperature regulation, but through behavioral means. And so when we think about heliothermy, sigmathermy, and kleptothermy, these are all mechanisms of thermoregulation that rely upon the behavior of the organism in order to maintain their body temperature, moving in and out of sun and shade, crawling under rocks, crawling down into burrows, etc. There's a herpetologist at UC Santa Cruz named Barry Sinervo, and I learned yesterday that he evidently has died. I haven't seen anything official, but I've just seen posts on social media from his students and former postdocs and things. And after I saw that he had died, I went and revisited his work. He's a highly influential herpetologist who in the 1990s, this is not going to be on the test, just information for you. He did a bunch of cutting edge work on behavioral ecology of lizards, and more recently has been involved in looking at what the consequences of climate change will be for lizard populations. And in as I was looking through information about him online, I saw that he had given a TED talk about 6 years ago at UC Santa Cruz. And at the beginning, so I clicked on it just to kind of satisfy my curiosity as to what he might be talking about. He was talking about the extinction of lizard populations and entire communities of lizards, in some cases due to climate change. And at the beginning of the talk, he said, you know, I want here at the beginning of the talk to kind of get you inside the skin of a lizard, to get ingrained in your head or inserted into your head kind of what it's like to be a lizard in a landscape faced with the challenge of thermoregulating. And that's kind of what the purpose of this section of the textbook is about, is to get you inside the skin of these organisms so that you can see, okay, here are the challenges of thermoregulation and how do they deal with it? And they deal with them primarily through behavioral means. So let me share my screen and we will begin. All right, can you guys see my screen? Yep, okay, good. So I'm actually on the wrong slide, as it turns out. So we ended last time with kleptothermy, which is exhibited by a sea crate that lives in the burrows where these pelagic birds live when they're on land. And you can see that the sea crate maintains its body temperature at a much higher and more constant temperature than it would be able to if it was either out on the bare sand or if it was in a burrow that is not occupied by a shearwater bird. And so this is kleptothermia because it's basically kind of stealing heat away from the shearwater, which is one of these pelagic birds that comes on shore to nest. So that's in an adult individual, but what is also striking is that here is a developing embryo, and what they've done is they've cut away the shell so that you can actually see the embryo developing. As it turns out, you can do this with a chicken egg. If you have a chicken egg that is fertilized, you can put it in a small cup, put a piece of cellophane over it, and you can actually watch the chick develop in a cup to a certain point. And what they did was around this egg, they changed the source of heat as to whether it was coming from one side or coming from the top or coming from the other side. And then they monitored the movement of the embryo within the egg. And what they found is that the animal tended to move its body around inside the egg even, such that its body was pressed up against the edge of the shell that was receiving the most radiant heat. When you shift the heat source to the left, the organism, the embryo, shifts their position to the left. This takes a number of days to accomplish this shift. And when you shift the heat source to the right, as the embryo gets bigger, it's able to facilitate these moves more quickly, and it shifts itself to the right to follow the heat source. So whether you're talking about behavioral thermoregulation in an adult animal or in an embryo, this is evidently one of those abilities that these organisms seem to develop at a very, very young stage of development in order to, once again, regulate body temperature. The warmer you are as an embryo, the faster you're going to develop because all of your metabolic reactions are just sped up. And so it behooves an embryo, if it can, to orient itself towards heat sources, if at all possible. So do you guys know what the set point is? Physiological set point. And this applies to temperature, but it also applies to salinity and all kinds of other things.
Like the crucial value that homeostasis like works around, basically. So there's like a certain range, a little bit under and a little bit over, but the set point is like the ideal value for the organism to function.
Yeah, that's exactly what it is. So the set point is this physiological value that an organism in attempting to achieve homeostasis, it's kind of the goal of homeostasis. The goal of homeostasis is to have a body temperature at a certain body temperature. The goal of an organism, if you want to think of it this way, is to have blood sugar levels at a certain level. As your blood sugar levels fall, what will happen is it insulin that increases blood sugar levels or decreases blood sugar levels? Now I have to remember my physiology. Anyway, the body will either dump insulin into the bloodstream or take insulin out of the bloodstream in order to mobilize sugar so that you have a relatively constant blood sugar level. And so the set point is that value of blood sugar that the body is attempting to achieve. And the thing about set points for temperature is that the set point can change. And the set point for temperature is actually governed by the hypothalamus. So deep in the brain, at the bottom of the brain, where your brain is very well insulated from outside temperatures for the most part, there are centers in the hypothalamus that basically measure the temperature of the hypothalamus. And that then is how they determine whether or not, for example, in us, we're going to ramp up metabolism to increase more metabolic heat or slow down metabolism to create less metabolic heat. And this set point for temperature can actually be fairly plastic. Do you guys know what I mean when I say that a trait is plastic? Plasticity. If you remember back in genetics, we probably talked about phenotypic plasticity. What were we talking about when we talked about phenotypic plasticity? Do you recall? This is not in the book. So if you're looking in the book for it, it's not there. This is just calling on information you should have received in another class to help you understand the concept.
It means there should be a lot of variation in how like the genes are expressed. So even though the genes may be like the same genotype, but there's a lot of variation in the phenotype.
Yeah, so phenotypic plasticity, a good formal definition of it is the same genotype expressing itself differently in different environments. So that's what phenotypic plasticity is. But then there's just a generalized plasticity, which is just how people refer to variation. So right now, for example, what is the temperature outside? It's cold, isn't it? Right? How cold is it? Weather app. It's 43 degrees outside. When you walk outside today, it's 43 degrees and it's rainy, which makes it feel even colder. But like me, I came to class today with a fleece on and a t-shirt underneath that, because 43 degrees just right now doesn't feel terribly cold to me. Why not?
Because you've been experiencing colder temperatures all winter.
Yeah, so I
think, oh, 43 degrees is not bad outside.
Sorry, Kiara, you're breaking up. I didn't catch the last part of that.
No, I just said you didn't think it was that bad outside right now because you've just been experiencing such cold temperatures.
Yeah, so this process of acclimation, so you can become acclimated to your environment. You become acclimatized to a particular environment. And so it got really cold. What was that, like 2 weeks ago? When it was down in the teens and 20s and even got below zero for a number of days, 0 Fahrenheit for a number of days. It was very, very cold, such that now 43 and rainy doesn't really feel like that much, because we've become acclimated to kind of what our body's expectation is for what constitutes as cold. Here in a week or so, when it warms back up, you guys are going to be running around in shorts and t-shirts in 60-degree weather because it's so warm outside. Well, it's because your body's set point for what is a tolerable temperature has changed. If you go through the rest of the summer and we get to next September or October, when the temperature drops to 60, what are you going to be doing?
Wearing jackets.
Yeah, you're going to be throwing on sweaters and long pants because all of a sudden it's cold. The temperature is the same. It's 60 degrees, but 60 degrees in March feels really warm and 60 degrees in September feels really cold, because we have been acclimated over the summer to warm temperatures. So 60 degrees in September feels really cold. And right now we've been acclimated to cold temperatures such that 60 degrees feels really warm. And so our physiology, our set points change in terms of what we're capable of tolerating in terms of the temperatures that we are in. The same thing happens to amphibians and reptiles. So they have a set point. That set point is ultimately set by the hypothalamus. And generally in herpetology, this is viewed as a range of temperatures, but that range can shift seasonally as organisms acclimate to changing conditions. And just in terms of kind of what, we don't usually refer to this as set point. We usually refer to this as T sub P, which is the preferred temperature range. And the way we measure preferred temperature is we basically put animals in a thermal gradient The thermal gradient is I make them out of metal flashing. I basically take a piece of wood, I put metal flashing in these channels that go along the length of the piece of wood, and then I put it in a refrigerated room, a very cold room, and I put heat lamps at one end of these channels. And then what that does is it sets up a gradient such that there are these channels that the animals are in, and they have a wire mesh lid so that the animal can't get out, but air can still get to the animals. And there are heat lamps at one end, and there's one of these downstairs in the basement in the animal room, and they're radiating heat on this end of the thermal gradient. There's a cold end because this is a cold room, and then you look and see where lizards are hanging out in this gradient. Some of them will be towards the middle, others of them will be more towards the hot end. Some of them will occasionally be right at the hot end. But over a period of about 4 hours, these animals are moving towards the heat and away from the heat as they behaviorally thermoregulate. And what you do is you put a bunch of animals into a thermal gradient, and then you come back, every 10 minutes for about 4 hours, and you record the temperatures of every animal in the thermal gradient, and you build a distribution of body temperatures. Sometimes you'll catch individuals that have been hanging out on the warm side and have just moved to the middle to cool off a little bit, so they'll be a little warmer. Sometimes you'll see individuals in the middle who have been hanging out on the cool end to cool down and are moving to the warm end to warm up, so they'll be a little bit cooler. But this middle 50 to 80% of values, 50 to 80% of values is considered the preferred temperature range. And the way to measure this is by putting these things in a thermal gradient where they could be anything from very, very hot to very, very cold, and then see where the animals themselves are choosing to hang out. In the old days, we had to actually calibrate the gradient itself, but now we can actually just take direct readings from the animals themselves in the thermal gradient. And so this preferred temperature range is kind of what the set point is. And in theory, if this preferred temperature range or the set point is subject to acclimation, we should see that set point shift somewhat seasonally. So if we collect animals like right now, like I'm going to be doing this week as I go to Texas and Oklahoma, we should see that their preferred temperatures will be cooler than if we went and collected those animals in August, for example. So if you look at what actually organisms do, is you find that when animals are cold, they will shift to warmer areas. When they find themselves heating up too much, they will then shift back to colder areas. And the goal of this shifting back and forth between these two environments is so that they maintain their body temperature at around this modal temperature of 39 degrees centigrade, which is a fairly warm body temperature. So the dashed line is the body temperatures of lizards. And if you find yourself down in this lower range of body temperatures, these animals are shuttling from cold areas to warm areas. And if they find themselves in this area, they find themselves shuttling the other direction. So the orange bars are the temperatures of individuals that move from a cold area to warm area or warm area to cold area. And then these are actually the observed body temperatures of lizards in this population. And so once again, they're achieving a relatively constant body temperature, but they're achieving it solely through behavioral means, moving into and out of cold and warm areas, depending on what their internal temperatures are. So as I said, it can change over time seasonally. People have also found that during reproduction, it changes. So tell me a type of snake that tends to brood its eggs internally and gives birth to live young? Go back to your snake knowledge. What are some groups that are viviparous? Can you guys hear me?
Boas and vipers.
What's that? Elizabeth?
Oh, boidae and viperidae.
Okay, so vipers are what I was thinking of. Viperidae and also the water snakes, the Natricidae, are also live bearers. Boas lay eggs, and I guess there are some species that are viviparous. But I think of groups that are almost solely viviparous as the Natricidae and the Viperidae. Anyway, these individuals are carrying their embryos inside of them for long periods of time during gestation. And one of the things that you will find is that they also thermoregulate at different temperatures. If you're brooding offspring, what kind of temperatures would you expect individuals to be brooding at cooler temperatures or warmer temperatures?
Warmer.
Yeah, generally warmer, because warm temperatures is going to speed up a rate of development of the embryos, going to shorten the amount of time that the female is carrying those offspring because they're developing faster. And that's actually good for her because, of course, the whole time she's carrying offspring, that represents an extra burden of weight on her. She slithers more slowly than she would if she wasn't burdened by that extra weight imposed upon her by reproduction. And so the faster she can develop those offspring and give birth to them, the less time she spends being vulnerable because she's carrying offspring. Similarly, what would you expect if an animal is infected? It turns out that that amphibians and reptiles are prone to all sorts of infections. I'm thinking of one condition in anole lizards that occurs fairly commonly in the tropics is there's a variation of malaria that lizards can get. And when these lizards are infected, what do you expect their set point to be, warmer or colder? What happens to you when you get an infection?
Also warmer.
Yeah, so once again, in the same way that you get an infection and you get a fever, that's your body's attempt to kill whatever it is that has infected you before the increase in temperature kills you also. But yeah, the set point will rise if you've been infected. So all of these things can alter the set point, alter the preferred temperatures at which you are thermoregulating. So I'm not sure why this figure is in here. This is a figure from your textbook. And what it's probably doing is it's showing you two animals that are thermoregulating at different temperatures. In this case, this happens to be a pit viper. You know that it's a pit viper because you can see that the tip of the nose here is cooler than the rest of the body. And you can see that, yeah, I'm not sure. I didn't review this. I should have reviewed this before I came to class today. But this snake is thermoregulating at a lower temperature. This snake is clearly thermoregulating at a higher temperature. They now make these cameras that allow you to measure the temperature of an organism using thermal imagery, which is something I've never used before. It's fairly expensive, but it's something that wasn't really available, 20 years ago when I might have been interested in doing some of this stuff. I'll bet those. This is a well snake. This is a sick snake. Now I'm recalling. These are thermal images of an infected snake and an uninfected snake. And this snake has been infected experimentally. And what you find is after you inject an animal with pathogen, their body temperature goes up, whereas it was low before. And then when you look at the number of days after an injection, if you were actually injected with something that infected you, these are the temperatures of individuals 1 day after injection versus 7 days after injection. And you can see that their set point has gone up because 7 days after injection, the overall temperature has shifted to the right towards higher temperatures. And so once again, this is a change in set point as a result of being infected by a pathogen. So we've talked a lot about behavioral ways of thermoregulating, but there are plenty of physiological mechanisms for thermoregulating that are employed by amphibians and reptiles. But this isn't physiological in the form of creating metabolic heat. This is physiological in terms of kind of changing how the distribution of heat is distributed around the body. And so classic cartoony sort of thing from my childhood, at least. You guys know what a Saint Bernard is? Saint Bernard, big bulky dogs. Do you know stereotypically kind of what they're known for? In terms of their working kind of dog, what kind of work do they do? And I'm thinking of a work in the Swiss Alps.
I always thought they were a rescue dog. So like if you look at cartoons of them, they have like the barrel around their neck.
And what's in the barrel?
I honestly have no idea.
Okay.
Probably like alcohol is what I imagine, but I
Oh, of
have no
course
idea.
there's alcohol. Saint Bernard cartoon. So let's look up a Saint Bernard cartoon. They always have the barrel, and inside the barrel is usually a little flask of whiskey. Happy hour, 5 to 7. I should just copy this and put it in the PowerPoints. So why would a Saint Bernard that is going to save you from an avalanche have a little barrel of whiskey around its neck?
Isn't alcohol supposed to raise your body temperature? But doesn't it not actually?
Okay, good. Okay, so explain that to me, Kelsey.
I don't know why it doesn't. I just know that I like heard that like, oh, drinking makes you warmer, but I thought that it wasn't true.
So drinking will make you temporarily feel warmer. Why does it make you feel warmer? Is alcohol a vasodilator or a vasoconstrictor?
Constrictor.
It's not a vasoconstrictor, it's a vasodilator. Do you know anybody in your family or any friends or anything that when they drink, they get red-faced and flushed? My partner's mom, if you give her a glass of wine, 3/4 into the glass of wine, she is red-faced as can be. And it's because alcohol is a vasodilator. And so when you drink alcohol because you're cold, you initially feel warmer because what's happening is your blood vessels of your skin are dilating. You're getting more blood flow to the surface of your skin. So the receptors, the sensory receptors that sense cold in your skin actually sense warmth because you're transferring blood from your core body, which is warm, to your skin, which is cold. That warms up your skin. But the bad thing about that is that while you feel warmer because you're vasodilating those surface capillaries, you're basically transferring heat from your core body to your skin. And so while you feel warmer, what is happening to your overall body temperature is your body temperature is cooling down, which is of course exactly what you don't want to do if you are actually in an avalanche and are at risk of freezing to death. You don't want to speed the process along. So Kelsey, you're right in that it has this reputation for making you feel warmer, but in reality, it's doing exactly the opposite of what you want to do. Because of course, when you're cold, your body vasoconstricts the capillaries at the arterioles and capillaries at the skin, which basically reduces blood flow to the skin, which then preserves core body temperature. And so simply by dilation of the blood vessels, so if you're getting too hot, you can dilate your blood vessels, increase your heart rate, that pushes blood out to the skin, and as a result, any heat that is in your core is then radiated out to the atmosphere, and so you can dissipate heat. In the same way, if you're cold, but then you have a portion of your body that is in the sunlight, you can basically, once again, through vasodilation, you can shunt blood to that area that is exposed to a warm surface or a source of radiant heat, warm up that blood, then as that blood circulates to the rest of the body, it will once again raise your body temperature. By either constricting and dilating blood vessels and altering heart rate and things like this, you can actually alter the flow of blood around the body such that you can heat up quickly or you can cool off more slowly, depending on how you are actually taking advantage of your circulatory system. So for example, countercurrent heat exchange is when you have, for example, a limb, and as blood flows out into the limb, it's going to cool off, and then as it returns, it's going to heat back up. And it heats back up before it comes back into the body, because as it's coming back into the body, it's right next to a blood vessel that is leaving the body. So this blood vessel leaving the body has nice warm blood in it. That blood is going to cool off as it goes out into an appendage, and then as it comes back, it's going to pass right by the same artery, such that this artery with nice warm blood coming from the core of the body is going to heat up the blood that is returning to the body. And once again, this helps conserve core body temperature, even though the body temperature in the appendages will be somewhat lower. So when you go to the doctor and you get your body temperature taken, traditionally, how did we do this before we had surface temperatures that they run across your forehead? And maybe you've always gotten your temperature done with a forehead thermometer.
Under the tongue?
Yeah, you would generally put a bulb thermometer under your tongue. Well, the reason that you put your bulb thermometer under your tongue, have you ever looked at the underside of your tongue? All of you have cameras on. Let's everybody look at the underside of our tongues. Let's see, let's get this big so I can see my own. They're disgusting. Why are they disgusting looking? Now I've totally grossed you guys all out. Oh, you're all turning off your cameras. Nice. Why does your tongue look so weird underneath there? The underside. They've all left the room. Are you guys still there?
Yeah, I just don't think any of us know the answer.
Oh, well, if you look in a mirror, when you turn your tongue up, one of the things that makes it look so weird is that you can see all of the veins and arteries and stuff running through the bottom of your tongue, and it just looks kind of weird and gross. Well, so when you put a bulb thermometer under your tongue, it's coming into very close contact with your circulatory system. Not only that, it's inside of your mouth. You know, when you go to the doctor and they take an oral thermometer reading of your temperature, they tell you to keep your mouth closed. So it's going to give you a measure that is much closer to your actual core body temperature than if you took it, for example, under your arm or at your fingertip or something like this. Those are all going to be cooler than if you took it through your mouth. Or the other way in which we used to take temperatures is by taking anal temperatures in reality. We don't do that anymore. Now they use that thermometer reader that goes across your forehead, but it's of course calibrated knowing that it's taking a skin reading rather than taking a core reading. So it's calibrated in such a way that it accounts for the fact that your skin is going to be a different temperature than your core body temperature. The unfortunate thing about those things is they're somewhat inaccurate because depending on kind of, how thin you are and things like this, that difference between skin temperature and core temperature is going to vary from individual to individual. So the core temperature is what you're ultimately trying to preserve. And one of the ways of preserving that is through things like countercurrent heat production. Now, in horned lizards, they can take advantage of this because they have a countercurrent mechanism in their throat. And when a horned lizard is basking in the early morning, it leaves its whole body underneath the soil, and it just pokes its head up above the soil into the sun. And because it can control the blood flow that's going to the head, it can shunt blood to the head and then close it off and then just recirculate the blood in the head until that blood has warmed up. And then it will open up its shunts and then direct that warm head blood down to the rest of the body. So it basically uses its head to warm up the blood and then shunts that to the body to then begin to warm up the body in the morning. So we can have these countercurrent heat exchanges, which is what we just saw here. But then the other thing that we can have is we can actually have metabolic heat production that occurs as a result of what we call muscular thermogenesis. When you get cold, you shiver. One of the reasons that you're shivering is it's your body attempting to generate more metabolic heat. Shivering is the action of all of these muscles contracting very quickly. And as a result, you create a little bit of energy. That little bit of energy hopefully raises your body temperature because you're feeling cold. If you ever watch bees in the morning as they're leaving a beehive, they won't fly away immediately. They'll hang out at the entrance to the beehive and they'll just vibrate their wings rapidly. They'll just sit there and buzz. And what they're doing is they have these big muscles in their thorax that operate the wings, and they're basically just sitting there flapping their wings, using those muscles to generate enough metabolic heat that their body temperature warms up that they can then go out and fly effectively. What happens in amphibians and reptiles in particular, is that big snakes like boids, this is a boa that actually lays eggs, is they will lay eggs and then they will wrap their body around the eggs, and then you can actually see their body, their entire body that they would normally use for locomotion, the muscles of the trunk of the body that they would use for locomotion. They're just sitting there and they're contracting and relaxing, contracting, relaxing, contracting, relaxing, contracting, relaxing. And that muscular action and the fact that they're coiled up in this heat conserving ball with the eggs inside of there, that creates enough metabolic heat that when the chamber that they're in gets cold, they basically go into this brooding behavior where they use muscular contractions to generate metabolic heat, raising their body temperature and increasing their metabolic rate in an effort to maintain the temperature of the eggs at a particular temperature. So their body temperature goes up, the clutch temperature goes up, which is much higher than what you would expect just based on the temperature of the chamber that they're in. So the animal doesn't do this metabolic heat production at all when the chamber that they're in is warm, because they don't have to, because they're warm and the eggs are warm because the chamber is warm. But as chamber temperature decreases, they try and maintain their egg temperature at high temperatures, and they do that by basically this muscular contraction that generates metabolic heat through what we call muscular thermogenesis. So they can regulate their body temperature through metabolic heat production, but they only do it at certain times, and it exacts a pretty high metabolic toll on them, and that their metabolic rate goes way up relative to resting conditions. And so it's costly to thermoregulate in this way. Boas are well known for doing what? Boas and pythons are well known for doing what in terms of their energy gain? Do they eat a lot and eat often, or do they eat less seldomly, less frequently?
Don't they eat less frequently?
Yeah, they tend to eat really big meals at very infrequent intervals. And one of the reasons is that they have a relatively low metabolic rate. And so they can eat a big meal, and then they can sit around for the next 3 months and not eat anything because they're not really burning up a lot of energy. And so they tend to eat big meals less frequently because they don't have to eat like we do, a series of smaller meals more frequently. The reason we have to eat smaller meals more frequently is because we're little metabolic powerhouses, because we're generating metabolic heat in an effort to keep our body temperature at a relatively high constant level. These snakes don't do that. So when these snakes engage in muscular thermogenesis in order to brood eggs, they're going to be consuming a lot more energy than they would normally be if they weren't doing this brooding behavior. So it's costly metabolically for them. Us every day is costly metabolically, which is why we eat 3 meals a day or 5 meals a day or however many meals a day that you eat. If you skip a meal, like if you have class during lunchtime, so you get breakfast and dinner, by dinner, you're pretty hungry. Well, you've been burning up energy all day in your normal activities, but a big chunk of that energy is just metabolic heat production. So we can think about amphibians and reptiles as either being more on the side of an active thermoregulator or more on the side of a thermoconformer. And so what do you think I mean when I talk about thermoregulators versus thermoconformers?
Thermoregulator.
Thermo.
Oh, sorry.
Sorry, Kelsey.
Go, Kelsey.
Thermoregulators try and like maintain their set point, like maintain it, whereas thermoconformers like fluctuate more with the environmental temperature.
Okay, so Quinn, when we read the Coles and Bogart paper, there was this contrast between nocturnal and diurnal organisms, and it used to be thought that nocturnal and diurnal organisms would be similar in their thermoregulation, but they found that they're not actually. So diurnal organisms and nocturnal organisms, which of those do you expect would be thermoregulators versus thermoconformers? Quinn.
I feel like nocturnal would be thermoconformers,
Why?
and then diurnal would be thermoregulators.
Why do you
Just
think that?
because I just like with nocturnal, like it's more environmental, just because the temperatures at night tend to, I feel like, range more drastically than temperatures during the day.
So temperatures during the day actually fluctuate more because you have more variation in solar radiation during the day. Temperatures at night are actually kind of more constant because you have a constant lack of of solar radiation. And so they're generally lower, but they're also less variable in a way, simply because there's not temporal variation in solar radiation because there's no solar radiation. But because there's no solar radiation, if you're a nocturnal organism, what opportunities do you have for behavioral thermoregulation? Still Quinn.
Not really sure.
Okay, anybody else? If you're a nocturnal organism, do you have more opportunities for behavioral thermoregulation, or do you have fewer opportunities for behavioral thermoregulation?
Fewer?
Yeah, fewer, simply because if you get cold, you can't just run out into a sunbeam and warm yourself up because there is no sunbeam to run to. And so one of the things that we find, generally speaking, is that diurnal organisms will tend more towards thermoregulation, and nocturnal organisms will tend more towards thermoconforming. So the way you figure this out is you ask yourself, what are the body temperatures of the organisms as they're actively thermoregulating? What would their body temperatures be if they were randomly distributed about the environment? What are the environmental temperatures? And then you ask yourself, how well do the body temperatures and the environmental temperatures match the set point of the organism? So if you know what the organism's preferred temperature is, if the environmental temperature is very close to the preferred temperature, the organism might maintain its preferred body temperature even though it's not actively thermoregulating. And so the body temperature may be close to the set temperature simply because the environmental temperature is close to the set point temperature. But if the environmental temperature is very different from the set temperature, but the body temperature is very similar to the set point temperature, that would be a sign that this animal is thermoregulating. So knowing all of these things about the organism is required in order to determine whether or not you are actively thermoregulating or are more thermoconforming. So this is an example with two species of lizards that I'm familiar with from Puerto Rico. The one on the right is Anolis gunlocki. It is a forest trunk anole. Anolis cristatellus, which is the one on the left here. This is an animal that is also a trunk anole, but it's generally found at lower elevations on the mountain, and it's also found in more open areas on the mountain. As a result, Anolis cristatellus lives in a more variable environment. And so the environment varies quite a lot. But this animal tends to thermoregulate around a relatively narrow range of its thermal preferences. And so we would think of this animal as a thermoregulator because it's maintaining its body temperature within this narrow range in an environment that is quite variable, and it sometimes falls very far outside of its normal range, of its preferred range. Anolis gunlocki also has a relatively narrow range, although you'll see that the preferred temperature range is wider than it is for Anolis cristatellus. But because it lives in a shady forest, its available temperatures tend to be relatively narrow in terms of their variation. So even though this animal is maintaining its body temperature within a relatively narrow range, just a couple of degrees centigrade, because that also happens to be the environmental temperature that it finds itself in, we wouldn't classify this animal as an actively thermoregulating organism because it's not doing anything active to modify its body temperature. Its body temperature is just taking on the body temperature of the environment that it finds itself in. And that's pretty typical of organisms that live in dense rainforests where they're in constant shade. They're generally regulating their body temperature at a lower body temperature, so down here a little above 25, as opposed to up near 28 to 29. It's a little more variable than an actively thermoregulating lizard, and it conforms to the range of temperatures that are found in the environment. That's characteristic of a thermoconformer, and it's different than a thermoregulator. And so once again, knowledge of the body temperature, the environmental temperatures, and what the preferred temperature is necessary in order to make this classification. So I have all this stuff in little things. So this just summarizes what I just said. So there's actually a calculator. You can measure the effectiveness of thermoregulation, where you look at the body temperature relative to the environmental temperature. You subtract that ratio from 1, and you see that Anolis crispitellus is a much more effective thermoregulator than Gunlockii is. And it's because their body temperature is very different from the environmental temperature that they find themselves in, as opposed to in Gunlockii, the body temperature is very similar to the environmental temperature where they find themselves in. So effective thermoregulation versus a not very effective thermoregulator. These individuals are thermoregulating, these individuals are thermoconforming. And so this is just a formal mathematical way of putting a number on that. When we look at body temperatures in geckos, so in this case, these are geckos of the desert Southwest. These are in the genus Coleonyx. These are ground-dwelling geckos. They do not climb walls. You generally find them under rocks and logs and things like this. They tend to be distributed throughout the desert Southwest, and you can see the different distributions. Coleonyx variegatus, Coleonyx brevis, which is the one that I'm familiar with from Texas. Coleonyx reticulatus is the Big Bend gecko. It has a narrow range within Texas, out in the Big Bend area of Texas. Coleonyx switaki. All of these individuals are in the same genus. But one of the things that has happened is that organisms that tend to live in desert regions have evolved two characteristics. The characteristics that they have evolved is they have relatively high active temperatures. So if you flip a rock and find a gecko under it in Southwest Texas, you're going to generally find that the body temperature of that individual is relatively high. Same thing, this is switaki, and even though it is not as closely related to these groups as it is to these more ancestral groups that live down in Central America, it has also evolved high activity temperature as well. Coleonyx reticulatus unfortunately has not, but one of the things about the Big Bend region of Texas is it's quite mountainous, and so there are plenty of coolish environments, even though this thing is embedded in the middle of a hot desert. So in these hot desert dwelling species, you find that they've both independently evolved high activity temperatures, but they also exhibit relatively low levels of evaporative water loss. And so something about their physiology has changed that enables them to withstand these high temperatures without becoming water stressed. These other species that live down here in South America that are largely forested, very mountainous, tend to occur at cooler temperatures. They tend to have lower body temperatures, and they tend to suffer more evaporative water loss. And so organisms have evolved physiological mechanisms that help them cope with high temperatures when they live in environments where the thermoregulatory challenges are a little more difficult to escape behaviorally. And so once again, as relative humidity goes up, evaporative water loss goes down. And these guys tend to aggregate, is the other thing about their biology. If you flip a rock in South Texas, you will tend to find a number of these under a single cover object. And the reason for that is that as group size increases, the evaporative water loss that they suffer tends to go down, and the relative humidity of the space in which you find them tends to go up. So this is a combination of being active at high temperatures and once again engaging in a behavior that reduces evaporative water loss in this case. Questions about thermoregulation? We're done with thermoregulation. This is the banded gecko from California. I think this is I think this is Cholionics variegatus. This one that lives over here, or fasciatus rather, used to be variegatus. Questions about thermoregulation? The next topic is energy relations. So if you go to your textbook, and I believe it's the next chapter in the order of chapters. Yeah, chapter 7, energetics and performance. You'll find that thermoregulation is tightly tied to energetics. The warmer you are, the more energy you're going to use, but it also influences things like your ability to locomote and things like this. And so these two chapters are relatively tied to one another. And so that's what we'll be talking about next time. So if you haven't started reading chapter 7 yet, please do so. And we will pick that up on Monday. Tomorrow is another spring break day, so don't expect faculty to be on campus or around tomorrow. We are under strict orders from the provost to take these spring break days off, as should you. I will not be in class on Friday because I will be in South Texas collecting geckos, and so we will reconvene on Monday. All right, I'm working on getting your exams and your literature summaries graded and back to you. Expect to receive them in the next day or two so that you have feedback from those two graded assignments. And I think that's about it for now. All right, if there are no questions, I'll see you on Monday.